Good.
Good afternoon. This is breakout session, the gut microbiome, what we know and where we're going. I'd like to introduce Dr. Hove, who is a senior consultant and professor of gastroenterology at the Department of Transplantation Medicine at Oslo University Hospital, and is leading the research group, Genomics and Metagenomics in Inflammatory Diseases. His current research focus is the role of gut microbiome in PSC and other inflammatory conditions with gastrointestinal manifestations. Dr. Hove is a member of the PSC Partners Scientific Medical Advisory Committee.
Okay, thank you very much for the introduction, and thank you very much for the invitation, for the opportunity to be here. I'm really grateful to PSC Partners for inviting me and also for funding some of the work I've been doing. So the idea of this session is really to talk about the microbiome. I will try to highlight, I'll give you a bit of a background because people may not know what's going on, and then on how and why we do microbiota research, and then I will highlight a few topics that I find specifically interesting. So I will not cover everything, but afterwards there will be plenty of opportunities to ask questions, and then we can cover everything if you want to. So I would just highlight that some of the things that I think is really interesting is whether specific bacteria could be driving PSC, and I will also focus a bit on the gut as a biochemical factory, which I hope you'll appreciate and agree with me. That's an important thing. And of course, for you and for me and for us, could this somehow help us to improve clinical practice? And I'll summarize in the end. So from a scientific perspective, we really don't know what's going on in PSC. And if you ask ourselves, so in any disease, more or less, so what's causing this disease? And they will say, it's very easy to say, well, it's a product of genes and environment. And of course, that's probably true, but doesn't necessarily help you a lot. It could be difficult to understand, but the idea is really that our genes is sort of the prescription of our bodies, while the environment then will be all the exposures shaping us while we grow and while we live. And for many diseases, we know some, but not much. And I think, for instance, actually characterizing environment is very difficult. I started out my career with genetics, and we know a lot about genetics in PSC. And if you think about diseases in general, you can think about them as mainly sort of gene-driven or mainly, let's say, environmental-driven. As just an example, let's say COVID infection. That's obviously a disease which is mainly driven by the environment, by COVID. There are actually some genes that may influence how easy it is to get the COVID disease, but still an infection is largely environmental, while there are clearly diseases where if you have a genetic variant, you will almost always get the disease, like cystic fibrosis if you have 2 of these gene copies, and then those are very much on this side. But actually our data on genetics in PSC show us that genetics is not the most important things. There are changes in the gene pool, susceptibility, as we say, but there are other factors that are clearly more important. And this means that to translate it, that in, say, in a family with a person with PSC, there is most often not another one. In some families there are more than one, but in the majority of families there's only one. But this is, how does the microbiome come into play here? And that's really, we need to ask ourselves what's going on in the environment. And then the big question here is, could we consider what's going on in our guts? The microbes living there as sort of as an environmental factor or as a link to the environment. Just to briefly introduce you to this idea, let's start with the terms here. I confuse these a bit, I mix and match, but commonly, if you define them strictly, you would say microbiota is like all the microbes in one environment. While if you talk about microbiome, which is a bit cooler, we all talk about omes these days. We talk about all the genes. But if you think about like the microbiota, microbiome on humans in the GI tract, it's all the way from our mouths to the rectum. You have microbes. The majority are bacteria, and the majority of these are in the large colon, or in the colon. Say roughly 100,000 billion bacteria. So that's really a huge amount, and obviously a potential source of something that could influence our health. In addition, you have lots of viruses, you have fungi, and we know less about them than about the bacteria. So I'll focus on bacteria here. These bacteria, they have many different genes, at least 10 million known different bacterial genes, which is, say, 500 times more than the gene pool of our humans. So this is really a source of variability. Why are we interested in this in PSC? And it's kind of obvious when you look at the disease. Up to 80% in the population of PSC, typically up to 80% will at some time point have a diagnosis of inflammatory bowel disease. While reversely in the population of IBD, let's say 5%, more or less, in an average population, depending on what you're studying, will have PSC. So why only 5% is actually a question in itself, making it very interesting to study IBD. But there's obviously a close link here, and I usually call PSC a disease of the gut-liver axis. And what does this mean? I try to highlight it here. So it's actually so that the entire intestine, small and large, all the blood from it goes into what we call the portal vein, a large vessel entering the liver. That means that everything going on in the gut, all we eat, with nutrients, with all bacterial products, will actually end up in the liver. So quite obvious, something going on from the gut to the liver. But this is not a one-way system. The liver produces things as well. This is shed in the bile, and actually bile has lots of potential to influence the environment and composition of the gut. So here you have really a bidirectional system where the gut can influence the liver and the liver influence the gut. But there is more to it. What's coming down here, that's the esophagus, so what we eat, the food, all drugs, everything will enter this gut-liver axis system. And this means that there are many factors that influences this that are difficult to get an overview of, but it's also sort of promising, but this means that we have access. We can study it, maybe we can manipulate it. I think it's important to remember also that you can have symptoms of liver disease also outside the liver. And what comes from the gut here via the liver could also spill into the blood and infect our other organs. So this is really the gut-liver axis where the microbiome may have relevance. How do we study this? When we study bacteria, the classic methodology is to grow them in dishes like this with very nutritious gels, and you get these colonies. But there are so many different bacteria in the gut, it's very difficult to sort of grow many of them. Many of them they're actually anaerobes. They don't like oxygen. There is very little oxygen in the colon, and they die and won't grow. So, the big advances here really started when we could study the microbiome using microbiomics, which is sort of modern genetics-based technology. We actually have to then extract DNA from bacteria and study the bacterial genes. And actually these genes, some of them are quite diverse. So if you know the composition of different variants of some of these genes in a sample, you could actually look up in databases and get an overview of all the bacteria present in the sample. So this is the standard methodology have been over at least a decade. And how will this look? Well, this is like the normal microbiota composition. This is actually from one healthy individual. This is one sample. It's like a fingerprint, 100% of the sample. One color could be one bacterium. So the wider the thing is, the more of it is in the sample. So this is, of course, interesting enough. Many different bacteria going on here. And you have many of these. Here is actually 117. You make this thing here very narrow and put them next to each other and sort according to this red one here. You can really see that's extremely variable. It goes from like 60 to 70% of this bacterium until here, in this sample, less than 10%. And these are all healthy individuals. And this highlights extreme variability. Remember that genetically, you and your neighbor here in this enormous hall is almost identical, right? They're almost identical. While your microbiomes in your guts probably less than 50% overlap. So if something going on here really relevant for your health, there are lots of opportunities for this to sort of to be specific for your individual. So this is something you have to take into account when you study this phenomenon. This is not likely to be the same for all. It's also actually a bit tricky, but because it means that when you compare two different groups, the variation within each group could be as big as between the groups. So this is not easy. So this is sort of, we have been studying PSC microbiota for, let's say, a decade now, and I would say this is the main conclusion. In stool samples and also in mucosal samples, biopsy samples from people with PSC, the microbiota composition is less diverse. So illustrated here by fewer different bacteria. And this is sort of very simplistic the way it is. So in healthy individuals, there are more different bacteria. And this is a phenomenon that we actually see in many different conditions. And we don't really know what this means. There are also specific bacteria that there are more of or less of, but this is less consistent from study to study, and I'm not really sure we have been able to pinpoint the most important ones yet. But the tricky thing here is, what does this mean? And the big question is it the diversity is low driving disease, or is it actually disease changing the environment of the gut, etc., leading to low diversity, which could then perhaps in the next run be bad for your liver? We're looking at the major chicken or hen and egg problem that we have to go into. And how should we study this? And our big questions are really, is it the gut microbiome that caused disease or initiated, or does it modify disease? So could it make it worse, or could it make it better? So both could be important to know more about. So how can we study sort of cause and effect? And this is where one place where you could study cause and effect are mouse models. Here's just one example. And here, there are mice actually that spontaneously develop disease of the bile ducts. This very happy mouse here is called NOD C3, C4. They have biliary disease, and if you compare it to a cousin here without biliary disease, the composition of the gut microbiota are actually quite different. The thing is, in this study here, we made these bile duct disease mice germ-free, and that is actually possible. And that's sort of the other major advance in microbiota research that made this possible. It's actually possible to render mice germ-free and maintain them germ-free. And what happened here, when these mice became germ-free, their biliary disease got milder. So, loss of microbes, milder disease, which is interesting. There are other mouse models with other mechanisms behind their biliary disease where the germ-free state makes them worse. So this makes it complex. The disease mechanisms sort of determine whether microbes are good or bad. But it at least highlights that there is clearly some effect. You can actually manipulate something in the gut and something will improve or worsen in your bile duct. So that's sort of promising in terms of cause and effect, which are important to know if you want to make good treatment. So what will be the aim? Where should we go here? What is the aim of microbiota research? I think it's quite evident from what I already said that we're still here where we really want to understand disease causes and mechanisms. But I would also highlight that, at least I believe and hope that the clinical application, so changes or things we can do in clinical practice could perhaps be not too far away when we understand more about cause and mechanisms. Potentially could microbes be relevant for, or their products, be relevant for diagnosing disease or for monitoring, predicting outcomes? Could it perhaps be some way of select for treatment? If you have this and this bacterium, could that mean that we could give this and this treatment? And could we actually treat the gut itself, either via drugs, even like antibiotics, or preferably, could we find a good diet that makes the good bacteria happy and improves the liver disease. So I will highlight a couple of observations that I think are of particular interest. And the first is this, what I will call the Klebsiella story. And I'll read the title here of this paper. 5 years ago it came out. It says, gut pathobiomes underlie intestinal barrier dysfunction and liver T helper 17 cell immune response in PSC. So what does this mean? Very complex title. But I think the idea here is that something in the gut does something to the, say, the gut barrier, and they seem to show that it influences the immunity, immune response, inflammation in PSC. But what is this pathobiome thing? And that's sort of thinking a bit, going back, instead of thinking of the entire microbiota as how the community is important, it's more like highlighting, could there be individual bacteria that if you have them, this makes disease worse? It's more like a classical infectious disease. You have a bacterium that makes an infection. So this is where sort of the pathobiome term comes into play. I'll talk you through it because it says a little about things we can do now to study microbiota. So the point is, in this study they did the first humanization experiments. That does not mean that the mice sort of get human features, but it means that we give them a human microbiota composition. Some mice here got healthy stool samples, actually healthy stool, they were given by oral, by tube, and some got microbiota from persons with PSC and ulcerative colitis. And then they followed these mice for 4 weeks, and I think it's notable here that they actually didn't get any liver disease. It's not like, I think that's a good thing, that the microbiota from PSC immediately gave them bile duct disease. But there was some signs of immune activation. So more immune cells active if you have this PSC microbiota. So something perhaps going on. But what they next did is that they did the same colonization, humanization of the mice, and then they gave the mice bile duct disease by giving something called DDC in the diet. And this is toxic to the bile ducts, and you get a biliary disease. And then the PSC microbiota was important. The mice with the PSC microbiota clearly got the worst disease of their bile ducts. And this, I think, really shows something going on in the PSC microbiota is actually relevant for the disease process. They also measured more endotoxin, that's bacterial products in blood. The question is, why did these mice get more severe disease? And in this paper, they were interested in different bacteria, these pathobiome hypotheses. And what they did is that they looked at lymph nodes that are nodes in your stomach that drain immune cells, but here they found that the bacteria grow. They grow from these lymph nodes. 3 different ones. One of them was Klebsiella pneumonia. There was one called Enterococcus gallinarum, and one's called Proteus something. And the thing is, if you had only these 3 bacteria in a mix and gave them to the mice behaved very similar to giving the entire PSC microbiota, suggesting that perhaps it's the effect of these 3 bacteria together that was important. If you only gave them one of the bacterium, it wasn't enough. And the question is this relevant in human PSC? Well, they did study some people with PSC, and they see, at least in their cohort, they found more Klebsiella than in the healthy individuals. And then they studied this a bit more in the lab. They put these bacteria on cells from the lining of the gut, and they saw that they could make small holes, so suggesting that perhaps are these bacteria making like a leaky gut, and you get these bacterial products over in blood, and this will influence the liver disease. So all in all, very interesting. I would say got a lot of attention. And it's a very simplified model. First, they think that something going on in the gut, this is the barrier, and maybe these Klebsiella then makes holes leading to leakage, which then leads to inflammation in the liver. So the questions we are left with is, they didn't really show that they induced any leakage in human PSC. And it's a big question, I would say, how common these bacteria actually are. But this we need to know more about. And I think we should, there is clearly some promise here. And what is really promising is that could it be so that if you have these specific bacteria, these could perhaps be a subgroup that could be targeted, perhaps could we treat this bacteria and this would improve disease. And there are promising ideas on how this could take place. There is something called bacteriophages. That is like viruses for bacteria. These viruses don't give disease to humans. They only attack bacteria, and typically they attack specific bacteria. So they generated a cocktail of bacteriophages that could attack this Klebsiella pneumonia bacterium that they were interested in. And they had this model set up again, and also with these disease-causing bacteria. And when they gave them these bacteriophages, the disease improved, suggesting that, at least in this mouse model, bacteriophages, viruses attacking bacteria, could be a treatment. So far, the use of bacteriophages for treating human disease is very limited, but this is actually an area of intense research. It brings me briefly into the gut barrier, because the gut barrier and so-called leaky gut was one of the first hypotheses related to the relationship between IBD and the liver disease. Could it be so that it's just like you have inflammation of the gut and you have a leaky barrier and bacteria sort of leaks over to the portal vein and induce an inflammation in the liver? However, and this is sort of a model of the gut barrier, and with this Klebsiella pneumonia hypothesis, you could envision that this actually takes place, leading to leakage. But actually, if you go through the literature, it's actually very limited evidence of this ongoing leakage in PSC. We need to study this a bit more, but I think in many of the mouse models we see there is lots of gut leakage, while in our cohorts or materials with biospecimen from people with PSC, we see not that much leakage. And this barrier is very complex, and there's lots of things to be done here. For instance, there's a mucus layer that's important. There are different barriers, and lots to be done, I think, still on the gut barrier. But so far, I'm really not sure this is where the primary damage takes place. So then to the other main question is, what are the bacteria actually doing? And we have a gut-liver axis here, but I think what's going on in the gut is really, it's like a biochemical factory. So remember, 100,000 billion bacteria, that's a massive metabolism. And this is really an integrated part of our own metabolism. It's not like first come humans and then the bacteria came there and started working. All along our development, all human cells have been exposed to sort of the byproducts of this human metabolism. So I think this is sort of a critical point in the thinking of how microbiome can induce liver disease. And just envision here, there are lots of bacteria. Everything we eat, also all drugs, meet this. And many byproducts, or actually products, are made. And I have read papers suggesting that maybe 10% of all molecules in our blood at any given time is actually bacterial. And the majority of these we do not know. But these will then continuously or episodically sort of hit the liver. And maybe this could be driving disease. Some could be good for the liver. Who knows? But this will probably go on all the time, and perhaps this could even be relevant for disease after transplantation. So what are metabolites? There are so many different things. The majority are really not known and difficult to study. So it's like sugars, look like this. Could be amino acids, proteins, looks like this maybe. Could be fatty acids, could be vitamins or other essential nutrients. And some could be like drugs, aspirin, and toxic substances like Roundup. These are all metabolites that sort of could influence the system and perhaps enter a portal vein and influence the liver. How can we study this then? That's the big thing. One thing is to study which different bacteria do we have. But here we have to know more about how they handle these substances. And then we need to know more about the bacterial genes. So we have to go back to the DNA and to our computers and high throughput sequencing. And if we study them, we did this some years ago, and then we found that if you're looking at the number of different genes, that's also reduced. Here we have healthy, and here we have PSC. So we have less different bacteria, but also less different functions. And we studied which functions are altered. And there could be so many. We found many, many functions that are altered. There are less different genes doing this and that. And I'll show you one example that we really highlighted, which is a gene which is called pyridoxal 5-phosphate synthase. It's the main gene producing active vitamin B6. And this is, it doesn't look very impressive here, but this is actually quite drastically reduced in PSC compared with healthy in our study. And so we have less genes, so there's less potential perhaps for producing vitamin B6, we thought. And just imagine if it would be like it, something, you eat something and there are metabolites in the food, and based on this, the microbiome will make something like vitamin B6. Well, perhaps this is so then that in the PSC gut, there are less bacteria making this vitamin B6. That's sort of our simple model. And we try to sort of validate this by measuring it in blood. And this is what we found. This is a picture where we see vitamin B6 concentration in blood. And each dot is one sample from one individual. And we had a cohort from Norway and one from Sweden. And here we have healthy individuals. And you can really see here that the median levels are so much higher in healthy than in people with PSC. And we typically say that around 20, which is here somewhere, is a threshold for clear deficiency. We still wonder whether this really is this relevant. Does it have any impact? And what we measured other things in blood then that are molecules that typically accumulate if you lack vitamin B6. And as you can see here, the lower vitamin B6, you really have an increase of these molecules, suggesting there is potentially a functional consequence if you have in deficiency. And the other thing suggesting this is relevant is that these are from the same individuals that we measured vitamin B6. This was done from a material sampled years ago, really highlighting the value of collecting samples and putting them in our biobanks. And you can see that the lower level you had of vitamin B6, the shorter time to liver transplantation or liver transplantation-free survival, suggesting that there is some association between the level of vitamin B6 and disease course. So the question is then, is supplementation of vitamin B6 helpful in PSC? And then I think that's a big question because you see deficiency of vitamin B6 in other diseases as well. So this is something that's disease specific. And there have been so many hopes for vitamins that that's sort of the cure or could help with many health issues. And often they have turned out to be disappointing. But I think based on our findings, we have a strong rationale for really testing whether this could be a good thing. So we have planned for a trial in Norway, and the protocol is sort of under evaluation, and I hope to start up probably in January. So we will know more, because we really want to know whether it's a good idea. We don't really want to add another pill to the pharmacy unless we have to, unless we know that it's for something good. Are there other therapeutic opportunities in the gut microbiome? I think one of the big topics in the field is fecal microbiota transplantation. You may have heard of it. It's typical something that gets some headlines, but for all practical purposes, it's about giving material from stool from healthy individuals to people with disease. And it could be prepared like this. You should know that about 60 grams of poop is necessary for 1 dose of a typical FMT treatment. And it has to be prepared, filtered, etc. And then it could be given via an oral route with a gastroscopy or with a colonoscopy. There's been one small trial from Boston by Allegretti. They gave 10 PSC patients FMT. And this changed their microbiomes. And then you can see their diversity increased, as you can see here. There were some changes in liver biochemistry, but such a trial is very difficult to evaluate. We can't really say anything sensible whether this is good for the disease, but Such trials are going on in many different diseases, and it's like a tool for studying whether microbiome is relevant. You know, if a fecal transplant helps out, actually seems to improve liver disease, then it really proves there's something going on in the gut. And perhaps we can use that as a window to understand what kind of treatment we should actually give. Because I don't really think that fecal microbiota transplantation is future treatment for most diseases, to be honest. It's hard to standardize. We need to know more what's actually going on, but it's a useful tool. And there are other trials under planning or even initiated with this type of methodology in PSC. So hopefully we'll know more soon. And then you have other things suggesting that something going on in the gut is relevant for PSC, because antibiotics change commonly used biomarkers like alkaline phosphatase. You have these data, this is metronidazole from data from Finland 2 decades ago, and you have studies, at least a couple of studies which are even controlled with vancomycin suggesting that it reduces this alkaline phosphatase. Unfortunately, so far we don't have data in control fashion showing that the liver disease actually, the progression is stopped or improved and so on. But there is a strong rationale for such studies which are sort of long awaited, just to know both to know if it works, but also to know how it works and why it works. Vancomycin has also potential role in there are some reports suggesting that it improves IBD, and there is some data suggesting it could also have specific effects on the immune system. So this is a promising thing, but in terms of sort of the clinical evidence that we usually require for initiating treatment in disease, it's so far not enough. We need more. I think I'm about to end this now, but I'll highlight a completely different type of study just to tell you a bit about how the gut microbiome and therapy field may be looking. This is a very interesting study dating back to 2015 now from Israel. In this group, they were interested in finding out whether there are microbial features and other things that determine your glucose regulation. And in particular, whether blood sugar increases after a meal. That's sort of a good measure of blood sugar control. And they had a big study of hundreds of people who had this sensor, blood sugar sensors, etc., implanted, and they got standardized meals and so on to measure the individual response to different types of food. And they measured everything with them: microbiota, genes, many different blood tests, other anthropometrics, etc. And then they made like an algorithm to really determine for the individual what is good for you, which food should you eat to have a good blood sugar and low or bad blood sugar. And the thing is, they made many interesting observations on the way here. And these are just examples from 2 different participants in this trial. So here is participant 445, who is giving a banana. And you can see, perhaps you've been outside to doing a workout, I don't know, whatever, and you feel, I need a banana to get the blood sugar up. And this participant here had a nice blood sugar increase as expected after this banana. However, this participant did have no response to banana whatsoever, while this specific cookie meal here had no effect in this participant, while it had a very nice increase of the blood sugar in this participant. And this could actually be predicted from their microbiota composition and so on. And this is not intuitive at all, because you expect sort of the blood sugar to increase when you eat something. And the point is, this has been sort of repeated. It's actually possible to predict the response to different types of food. And this could even perhaps improve your blood sugar control if you have diabetes. But the point with this is really to show that if we're going to treat the microbiome, it's unlikely to be one therapy for all. And this we also have to take into account when we study microbiome in a disease like PSC, that perhaps are we looking for the subgroups, and those are the ones that we have to identify and target. There are many other things relevant that you can perhaps potentially go back to when you ask questions afterwards, but to really summarize this, I think I believe in this, that in some of people with PSC, there could be particular bacteria that may be driving disease. So that could be one mechanism. And but I also think that the microbiome has a really altered potential for metabolism of many essential nutrients, like vitamin B6. And this could actually be a disease relevant and potentially be driving disease or protecting against disease. But I think there are many different mechanisms like it. There's not like one feature fits all. And I think the fact that this is accessible makes it possible for us to foresee that there could be a room for gut-targeted treatment in PSC. But we are not there yet, so I'm an optimist, but I'm a bit of a cautious optimist. And I guess that's what I had to say. This is my group, and I'd like to thank everyone who has been helping supporting my research, including PSC partners. So thank you very much, and I'm happy to take any question. You should probably go to the microphone for questions. So I would just suggest lining up, and I'll try to point in the direction of the microphones. And then we'll just keep on going until time is up. So go ahead.
Can you hear me? Can you hear me?
Yes, I can hear you. I think.
Okay, I was wondering if you know of any studies regarding PSC patients in all different countries. Do they have a higher rate of PSC with IBD in certain countries, or it's across the board?
I think that's a good question. I think there is sort of the evidence here is limited by the number of studies and so on. But overall, it seems like, for instance, in Italy, there could be, or in Southern Europe, perhaps less IBD in PSC, and also in Japan. But so I think it's not unlikely that there is variation in the degree of IBD in different countries. But the reason for this, I think we do not know. It's really cool.
Would the gut microbiome look the same in PSC patients that didn't have IBD as the ones who did have IBD?
That's also a very good question. So that's sort of one of the biggest topics that we look. That's the first thing we look for after studying, comparing PSC with healthy. We would typically look for those with and without IBD. And surprisingly, it's extremely similar, at least when we look at fecal samples. I think in many studies there have been quite consistent, few or no differences in those with and without IBD for some reason. And I've been interpreting this a bit like, could it actually be then that it's the liver disease that is shaping the composition of the microbiota and not the other way around? But we really don't know. But when we have recently looked at the lining of the intestines or the mucosa, and there you could see a bit more differences in those who have inflammatory bowel disease and those who have not. And that is perhaps not surprising, because if you actually have an inflamed wall, you have less mucus and so on, and there will be other bacteria attaching to the epithelium. So some differences there are, but sort of in the fecal sample, there is not so much. And it also highlights a bit of the limitations with fecal samples. They probably reflect mostly what's going on in the last part of the gut, while there could be many things going on further up, and also in the small intestine, where sort of the number of bacteria are less, so it will be difficult to sort of to find differences in the small intestine by looking in stool.
What is the risk if you decide to join a trial, or even decide to just say take vitamin B6 or whatever, that then you'll be disqualified from some other great trial that comes up, or for some treatment because, oh, you had that thing, we want all the samples the same, and you can't join anymore?
Well, so you ask about whether the risk for losing out on other things, or the risk of actually being part of the trial.
I couldn't hear that.
So the way you wonder, so if you enroll in a trial,
Yeah.
whether you cannot be get other treatments and things like this.
Like even permanently? Like,
Oh.
you know, you decided on this trial, that kind of sets your track for years.
In general, if you're part of a trial, it's unusual to be included in another trial. However, I think if a drug becomes available, meaning that it's now clinically, it's sort of state of the art to give such a treatment, this would have to sort of be taken care of in such a study. So meaning that they would have to find some way to give people the treatment that they should have, even though they are in a trial. But but usually you have an idea which drugs will become available the next year or two. So I think in practice that is not a big problem. But I think it's unusual to be part of several trials at the same time.
Hi. I'm curious whether if someone has an intervention on their gut microbiome, like through a FMT or antibiotics or something, do they tend to return to this original microbiome, or do they tend to just, yeah, I'm curious about that.
Well, this is also one of the big topics of fecal microbiota transplantation, or how stable is sort of your gut composition. And this fecal transplantation thing is the really established indication clinically is typically an infectious disease called C. diff, we typically it's abbreviated, where you have one bug that is sort of taking control over the composition. So you have a major change in the composition in the gut, and it leads to colitis, which could be very severe. And if you give these fecal transplantation, it sort of turns off the infection, and things are sort of good. If you have a more normal, even though it's like disease associated and a dysbiosis, you could even call it, this type of microbiota are typically more resilient. So there are trials, for instance, where they have given actually fecal transplants from lean people to obese people that have type 2 diabetes, which actually turns out to improve glucose regulation, which is very interesting. And the effect, they could see it after 6 weeks, but after 18 weeks, it was no longer visible. And I think the majority of these studies now do repeated therapies. So it's a bit unlikely to see, at least that one or a couple of shots of FMT is sort of curing the disease for good. But this is sort of one of the big topics. How should we administer this?
And a quick follow-up, do colonoscopies tend to affect the gut microbiome?
Sorry?
Do colonoscopies tend to affect the gut microbiome?
If it affects, well, whether the scopy itself does it, I think, but of course the cleansing in advance of a colonoscopy is likely to have some impact. It is, there is evidence suggesting that. Whether it has a long-lasting impact is a bit hard to say, but you know, we have all heard reports of people having a colonoscopy and then this sort of, in some people with IBD, sort of their bowel habits changes a bit afterwards, for instance, and then this could potentially be related to microbiota composition.
Hi, could you talk a little bit about the priorities of your research and your lab team, and what expectations you're looking to, or you know?
So the priorities of my team. Well, I think we should think that there are many possibilities here, but as we sort of follow the biochemical factory hypothesis as an important thing, because I believe in sort of the whole community, it turns out it's a bit difficult really to understand this very complex and variable composition of the bacteria, while if you more study the output, actually looking for these metabolites in blood that could be bacterial, then it's sort of a bit easier to identify signals from the gut that could potentially drive liver disease. So we are actively screening for these with sort of large-scale big data type of analysis, and we try to identify those that we think are microbial and drive disease, and then we test them in mice. So I would say we have promising molecules that we see in human PSC that also seem to change disease in mice. So hopefully we'll be able to show more of these data in not so long. But so that's sort of the main one important thing. The other, I would say, main topic of my group is to look in early IBD and to study microbiome, and then we try to see who develops PSC in the long run by doing MRI, etc. So these are sort of the two main themes.
Do probiotics improve the microbiome of the gut? And if yes, what microbes specifically should you look for in probiotics?
Yes, this is a very common question. We have done some probiotic studies in other conditions, and in sometimes we have been able to pick up a sign of the bugs in the stool, but not always. I think the probiotics are not really designed to colonize and stay in the gut. They pass through more or less, and you need to give them repeatedly. And I think in very few conditions there is any good evidence that the probiotics available now have any meaningful impact on the disease. So when I get this question, because many use and try probiotics, but from my point of view, I don't really know if it's good or bad even, but I think it's unlikely that they're bad. So I usually say that if you use it and you feel that this has a significant impact on your health and symptoms, then it's probably okay. But I find no reason to use probiotics because just out of a belief that this is good for your health. So that's one thing. The other thing is there are sort of second generation probiotics based on these new technologies and isolation of perhaps more interesting and active bacteria. There are other bacteria like Fecalibacterium prasnitzi, which is sort of a popular, potentially nice bug, is being tested in some conditions. The problem with these is often they're difficult to grow, they don't like oxygen and things like this, but there could be probiotics on the market in not too far future that could be of relevance in different diseases. But I would really like to see trials, of course.
Hi, I was just wondering if there have been any studies on the foodborne pathogens and their impact on the gut microbiome.
Well, on foodborne pathogens on the microbiome, well, I think I'm not sure if I know, but I assume that sort of if you get a foodborne disease, typically then it will give you abdominal symptoms, enteritis, so on and so on. That's very likely to, at least while you do it. While you have it, that sort of will have a big impact. It's actually, that's happened several times that we have, for instance, studied some immunodeficiencies, and we look in the stool samples and there are suddenly some, I would say, crazy results, and we go back and realize that because we find some Campylobacter, for instance, at the time of the sampling, they didn't have symptoms, but later on they developed symptoms. So we can sort of see signs of infectious disease in the gut. And the question is, perhaps what you're wondering is whether this has long-term consequences. And of course, that's very difficult to study because you didn't have a sample in advance before their foodborne disease. So it's a bit hard to compare. But of course, you have some instances of IBS, or irritable bowel syndromes, that are caused by infectious by microbes. For instance, in Norway, we had some years ago a large outbreak of Giardia lamblia, which is a parasite. And after that, there was a lot of IBS. And there has been studies sort of on the microbiome of these. And I think there are sort of changes. There are some changes related to irritable bowel syndrome in those who had this Giardia parasite. But I'm not sure. Yeah, so there's the one potential cause of these symptoms, but I'm not sure we know.
One more. What are the chances? It seems like the research is all over the place and still very early. that there is some magic thing, the bacteria that you didn't know, somebody figures out this is it, and the whole thing goes away with just taking a daily pill or something?
I think that the disease itself is so complex with so much variability and so on that it's unlikely that you will find one, this is it, and cure everyone. But I think we will have within I would say very soon sort of some ideas on which thing we need to test. But I think sort of the weakness here is there is still, we have the idea of something that could be helpful till you have a treatment, that will take some time. But if you think about vitamin B6, that could be very short, because I think if vitamin B6 deficiency is very common, and yeah, we don't know, we don't need very much evidence of a hint of an effect before so kind of supplementation with vitamin B6 would be a good idea to advise. But it's perhaps unlikely that that would be a sort of a cure, but sort of a positive thing maybe. I think the best chances of a rapid cure if you find a disease mechanism where you have a potential drug already. So because then it's repurposing of existing drugs, and those processes are a lot faster than sort of inventing everything from the bottom. For instance, if you have a process that will take a very long time is that if you find a molecule produced by an enzyme in the bacteria, and actually there are some examples where you can perhaps drug the bacterium making that metabolite. You don't have to kill the bacteria. You don't have to treat the body. You can actually inhibit the mechanism in the bug. But then there are so many processes until such a treatment will be available. So that will take a bit longer time. So I can't give you the correct answer, but I think I can only say that we work as hard as possible and try to sort of design our research in a way that it could be clinically relevant as soon as possible.
Does the gut microbiome change as a function of position in the colon or along its length?
Could you repeat that, please?
Does the microbiome change according to its location in the gut?
Oh.
I assume you're looking at fecal samples.
Well, from fecal samples we can't tell, but we have studied the gut mucosa. So we have a set of people going under colonoscopy, and we have then sampled from the end of the small intestine, terminal ileum, and several places in the colon. And actually, there's actually very little difference in the location in the colon, which was a bit surprise to us, because it's typically said that it's in IBD, PSC with IBD, you see more inflammation on the right side of the colon. So we were looking very hard on the right side, but there was not much to find there. So, but you could still envision that it doesn't have to be the way that it's actually in the colon that's important, even though there we see the inflammation. It could be further up in the small intestine. There are several meters of things that are largely a black box to us. There are some cool tools now where you can perhaps sample with a capsule sort of along the way of the small intestine, but it's still a bit ahead to use that as a tool to study the location. But I think it could be relevant potentially. People seem to run out of questions. We still have a few minutes left. So just things that I haven't mentioned that I could just briefly mention is that there is a lot of interest in the microbiota in bile. And it's a big question whether bile, if you're healthy, whether it's actually sterile. It's very difficult to prove that something is sterile because the methods that we use with microbiota studies is so sensitive. So you'll always find some bugs in there, but sometimes it could just be from contaminants in your lab liquids. But in PSC, there is actually lots, in many, 2/3 at least, we find bacteria in the bile. And the reason for this is a bit uncertain, but it seems like, in particular, if you can grow bacteria in the bile, like culture new grow like enterococcus or things like this, that's probably not the positive thing, suggesting that if you have colonized with bacteria in the bile duct, that sort of could be contributing to disease progression somehow, which is not surprising, but it's something that we could potentially attack then.
Studies have shown that there's less microbiota in the PSC patients. Does it seem that disease is causing less microbiota? Less microbiota in there?
So this is one of the big topics. I think it's because we see less diversity in so many diseases. There are, in some diseases, it's been possible sort of to have a sample before disease onset, and there could be some signs of low diversity before disease. And low diversity may be a bad thing, but we don't have any proof in PSC. But sort of in the field by itself, sort of the way we live now, in the Western world particularly, there is overall perhaps we're losing diversity. There could be important functions, important bacteria that are lost because of the way we eat, the way our food is made, the antibiotics we use, and so on. And this is sort of a worry that it is actually contributing to diseases. It's a bit on the theoretical, but it's an interesting hypothesis.
Hello? There we go. From a nutrition or dietary standpoint, are there items in a normal diet that manipulate the microbiome in either a positive way or a negative way? If you take celiac disease, there is a correlation with gluten. Are there any kinds of markers or indicators in nutrition and diet that would impact microbiome?
I haven't talked about diet so far. It's sort of the difficult topic, but I still think is one of the things that is so understudied and perhaps could be extremely important both for health, disease, and also for the effect of therapies. Recently, there was a small German study with gluten-free diet in PSC. It wasn't really designed to see any effect on the liver, at least they didn't see any effect on the liver disease, but they sort of see changes in the gut in those who get the gluten-free diet. And I know they are running now a larger trial over longer term in people with PSC on the gluten-free diet. So it will be interesting to see. But I think in general, if we can find a dietary solution, that would be great. You ask about whether I now can see this is good and this is bad. I cannot really do that. But so I usually recommend, I say like public dietary advice is good. Try to have enough fiber, etc. That is sort of the best we can do based on what we think about how fibers are important for, say, short-chain fatty acid production and things that we believe are healthy. I think perhaps the final question.
I wanted to ask you about fermented foods, just on this subject, fermented foods. So kefir and sauerkraut, from what I've read, it's been shown, and this may not be true, but it can increase the biodiversity of the microbiome.
That's a good question. Fermented food is popular. There is some, I can't really tell you whether this has a health benefit and so on. I don't have the full overview of that. But in general, I don't worry that much about. I think a varied, hopefully fairly nutritious food with enough fiber is probably a good thing. And really going to any extremes, there is no evidence, and you risk ending up more in problems. But there are situations where you sort of you worsen your symptoms from some food, and that would sort of motivate different diets and so on. And I think that is okay. But I don't think I can say anything specifically about fermented food, unfortunately. Okay, perhaps this is the end. Thank you so much for coming and for all the. There is a final question. Should we allow that?
Okay, so in the Nakamoto paper, there's a 3 mix of bacteria. Have they focused any effort on bacteriophage therapy for either of the other 2 bacteria, like E. gallinarum as opposed to just Klebsiella pneumoniae?
I'm not sure I really got the question, but I think we can take that afterwards.
Okay.
Thank you so much for coming, everyone. Thank you for all the questions, and have a good one.